So in the question it is asked to write the sequence of steps involved in the sequencing of genome of an organism. So it means we have to write the methodologies of the human genome project. So there were mainly three methodologies of human genome project. These were PCR, which is called as the polymerase chain reaction. Second one is the sequence annotation. And the third methodologies which is used in the human genome project is EST, which is also called as expressed sequence tag. Now before studying about the sequencing of organism, first we have to see what is the host and the vector. So host is usually bacteria or yeast like Saccharomyces or bacteria. There are many bacteria of the group Archaebacteria and Eubacteria. Anyone can be the host. And for vectors we have BAC, which is also called as the bacterial artificial chromosome, and YAC, which is called as the yeast artificial chromosome. These are the vectors which are used for the sequencing in the human genome project. Now let's first see about the first method, which is sequence annotation. So for sequence annotation, let's suppose there is a bacteria. So if it is a new species of bacteria, so what we will try to learn about this species, we will try to learn about the features like the morphology, structure of cell, so for knowing these for knowing this feature we have to extract the DNA of that bacteria. So by extracting the DNA we can know the sequence of the nucleotides like A, G that are present in that DNA. Example, I am giving an example of a nucleotide sequence that are complementary to each other. So this technique of sequencing is invented by Dr. Sanger, which is also called as the Sanger's method. So in brief we can say that first we have to find the sequence or annotate it, then we have to find the function of that DNA. So in definition we can write it as sequence annotation is defined as sequencing the whole set of genome containing coding and non-coding regions. And later assigning different regions in the sequence with the specific function. Now we will see about the ESTs, which are also called as the express sequence tag. Now we will see what is EST, which is also called as express sequence tag. So suppose this is a eukaryotic cell and this is nucleus, and within this nucleus there is chromatin, and this all is cytoplasm. In this cytoplasm there are certain proteins. These proteins are formed by the cell proteins that are formed by mRNA through the process of translation. So for in the process of express sequence tag, first we will extract the mRNA that is used to synthesize these proteins and will convert this mRNA into DNA by the process of reverse transcription. Suppose this is the mRNA. Now this mRNA will be converted to DNA, which is called as complementary DNA or cDNA by the process of reverse transcription using the enzyme reverse transcriptase. Now the DNA which is formed will fuse with this RNA because both are complementary, so they will fuse with each other to form a double strand. This double strand will be formed in which they will be connected by hydrogen bonds. Now we will use the RNase enzyme. The RNase enzyme will break the RNA and only then DNA will be left. So by express sequence tagging, we have identified the gene that was responsible for the formation of these proteins. This is the gene that was responsible for the formation of the protein. Now we can convert this into double-stranded with the help of the enzyme DNA polymerase to form a double-stranded DNA. by DNA polymerase. Double-stranded DNA. So by definition we can write EST means identifying all genes that are expressed as RNA, expressed as mRNA, and then as proteins. So these were the two methods for the sequence annotation and express sequence tag. These were the two methods for the sequencing of genome of an organism. The PCR will be studied in the chapter of the biotechnology. Thank you.